Today we shall discuss mineral nutrition. Mineral nutrients are essential chemical elements absorbed from the soil in the form of inorganic ions. Mineral nutrients are absorbed by the roots and then are translocated to various parts of the plant. Mineral nutrients are used in numerous biological functions. Mycorrhizal fungi and nitrogen-fixing bacteria often participate with roots in the acquisition of nutrients, as we will see later on. An essential element is one whose absence prevents a plant from completing its life cycle. It is a part of some essential plant constituent or metabolite and one that has a clear physiological role. Essential elements are composed of two types: the macronutrients, which are required in large amounts, and the micronutrients, which are required in relatively small quantities. Shown in this table are the essential nutrients in plants. There are actually 9 macronutrients required by plants and 8 micronutrients required by plants. In this table, the available form to plants are also indicated, and their major functions are also indicated. The first three elements, these are carbon, oxygen, and hydrogen, are not considered mineral nutrients because they are obtained primarily from water or oxygen. How about beneficial elements? How are they different from essential elements? Beneficial elements are elements that stimulate growth but are not necessarily essential, or they are essential only to a certain group of plant species. For example, sodium. Sodium is a requirement for halophytes, or plants that grow in extremely salty environments. Included in beneficial elements are sodium, silicon, selenium, and cobalt. Hydroponics is a soilless technique of growing plants with their roots immersed in nutrient solution. It is a technique to demonstrate essentiality of an element. In this technique, the plants are grown under experimental conditions, and one element can be removed at a time, and the effect is observed to determine its essentiality and to know the function of the element. The essentiality of an element will be harder to determine if plants are grown in the soil, since it is a complex medium which contains many different mineral nutrients. Let us consider the requirements in hydroponics. One is the maintenance of nutrient concentration. The nutrient serves as food of the plant and thus has to be replenished regularly. In the setups used in the lab using beer bottles, these are changed weekly to ensure that the plants have adequate supply of food. Maintenance of pH is another requirement. Shown in this figure are the mineral nutrients and their availability as influenced by soil pH. The thickness of the lines indicates the degree of nutrient availability to the plant root. As can be seen, all of these nutrients are available in the pH range of 5.5 to 6.5, as reflected by the thickness of the lines. Unlike at a higher pH, for example 7.5, where some nutrients or some elements are available, like molybdenum, for example, calcium, sulfur, while others are extremely deficient, like boron, manganese, and phosphorus. Aside from maintenance of nutrient concentration and pH, another requirement in hydroponics is sufficient supply of oxygen. In this figure, the radial path of ion movement from the soil solution through the root, that is, from the epidermis through the stele, is shown. As indicated in this figure, mineral nutrients are actively transported across the plasma membrane. As you very well know, active transport, which is movement against concentration gradients, utilizes ATP. More ATP is produced with aerobic respiration than anaerobic respiration. Thus, insufficient oxygen reduces nutrient uptake. Hydroponics is used in the commercial production of many greenhouse crops. In the hydroponic system, the roots are immersed in nutrient solution and air is bubbled through the solution. In another form of hydroponic culture, plant roots lie on the surface of a trough and the nutrient solutions flow in a thin layer along the trough over the roots. This nutrient film growth system ensures that the roots receive an ample supply of oxygen. In another alternative, which is sometimes heralded as the medium of the future, is to grow the plants aerophonically. In this technique, plants are grown with their roots suspended in air while being sprayed continuously with nutrient solution by means of a motor-driven rotor which generates the mist. This approach provides easy manipulation of the gaseous environment around the root, but it requires higher levels of nutrient than hydroponic culture does to sustain rapid plant growth. For this reason and other technical difficulties, the use of aeroponics is not widespread. The essential elements have three possible functions. They could either become part of the plant structure, they could play a role in plant metabolism, or osmoregulation. A particular mineral, such as magnesium, can be omitted from the culture medium to test whether it is essential to the plant. If the element deleted from the mineral solution is an essential nutrient, then the incomplete medium will cause plants to become abnormal in appearance compared with controls grown on a complete mineral medium. The common symptoms of mineral deficiency are stunted growth and discolored leaves. The symptoms of a mineral deficiency depend partly on the function of that nutrient in the plant. For example, a deficiency of magnesium, an ingredient of chlorophyll, causes yellowing of the leaves or chlorosis. In some cases, the relationship between the mineral deficiency and its symptoms is less direct. For instance, iron deficiency can cause chlorosis, even though chlorophyll contains no iron, because this metal is required as a cofactor in one of the steps of chlorophyll synthesis. Mineral deficiencies are more easily studied in hydroponic culture than soil-grown plants. Why? Because deficiencies of several elements may occur simultaneously in the soil. Also, deficiencies or excessive amounts of one element may actually induce deficiencies or excessive accumulations of another. In addition to this, virus-induced plant diseases may produce symptoms similar to those of nutrient deficiencies. That's why it is important that at the start of the experiment, only healthy plants or healthy seedlings will be used as starting materials. Mineral deficiency symptoms depend also on the mobility of the nutrient within the plant. If a nutrient moves about freely from one part of a plant to another, then symptoms of the deficiency will appear first in older organs. Young growing tissues have more drawing power than old tissues for nutrients in short supply. For example, a shortage of magnesium, which is a mobile element, will lead to chlorosis first in older leaves. On the other hand, if a nutrient is relatively immobile, like calcium for example, then a deficiency in calcium will affect young parts of the plant first. Older tissues may have adequate supplies which they retain during periods of shortage. In the example shown below, the plant is still healthy on the left. In the middle figure, the deficiency symptoms begin to appear. It is essential to catch the first sign of deficiency symptoms to determine whether the element causing the deficiency symptom is mobile or immobile. In the last figure, the deficiency symptom, which is yellowing of the leaves, or chlorosis, has progressed to other leaves. Based on what you see, is the element causing yellowing of leaves mobile or immobile? Let us consider now the functions of the essential elements, starting first with the macronutrients. Among the micronutrients, nitrogen is required in greatest amounts. In plants, nitrogen is available in the form of nitrate ions and ammonium ions. Nitrogen is a constituent of amino acids. It's part of the structure actually of amino acids, of certain hormones like auxin. It includes a nitrogen also in its structure and another hormone which is cytokinin. Okay. Also, chlorophyll molecule includes in its structure also the nitrogen. Also, the nitrogenous bases, pyrimidines, and the purines, which are nucleotide components of DNA and RNA. Nitrogen serves as a constituent of many plant cell components, including amino acids and nucleic acids. Therefore, nitrogen deficiency rapidly inhibits plant growth. Auxin and cytokinin induce growth through stem elongation and cell division respectively. Thus, nitrogen deficiency contributes to lesser auxin and cytokinin and lead to stunted growth. When nitrogen deficiency develops slowly, plants may have markedly slender and often woody stems. This woodiness may be due to a buildup of excess carbohydrates that can't be used in the synthesis of amino acids or other nitrogen compounds. Carbohydrates not used in nitrogen metabolism may also be used in anthocyanin synthesis, leading to accumulation of that pigment. This condition is revealed as a purple coloration in leaves, petioles, and stems of some nitrogen-deficient plants such as tomato and certain varieties of corn. Chlorosis is also observed when there is nitrogen deficiency because, as mentioned, nitrogen is a part of the chlorophyll molecule structure. Sulfur is absorbed by the plants in the form of sulfate ions. Sulfur forms part of the structure of two amino acids, methionine and cysteine. Sulfur is also a constituent of vitamins, biotin, and thiamine, and of coenzyme A, an important component in respiration, like acetyl coenzyme A and succinyl coenzyme A. Pyridoxine is a small protein containing iron and sulfur atoms, which is important in electron transfer reactions of photosynthesis. Many of the symptoms of sulfur deficiency are similar to those of nitrogen deficiency, including chlorosis, anthocyanin accumulation, and stunting of growth. This similarity is not surprising, since sulfur and nitrogen are both constituents of proteins. However, the chlorosis caused by sulfur deficiency generally arises initially in young mature leaves rather than in the old leaves as in nitrogen deficiency because unlike nitrogen, sulfur is an immobile element. Also, chlorosis resulting from sulfur deficiency is due to reduced protein synthesis rather than a direct impairment of chlorophyll synthesis. Chlorophyll is stabilized by binding to protein in the chloroplast membranes. With impaired protein synthesis, the ability to form stable chlorophyll protein complexes is also impaired. Phosphorus is absorbed by the plants in two forms more readily as hydrogen dihydrogen phosphate and less readily as hydrogen phosphate. Phosphorus is a component of important compounds of the cell. One example is the sugar phosphate intermediates of respiration and photosynthesis. Shown in this figure is the Calvin cycle where you have several sugar phosphate intermediates like ribulose biphosphate. One, you also have 3-phosphoglycerate, 1,3-biphosphoglycerate, and glyceraldehyde, thus 3-phosphate. Phosphorus is also a component of phospholipids and ATP and nucleic acids. Phosphorus deficiency results in stunted growth, presence of necrotic spots, dark green coloration of the leaves, production of excess anthocyanin, production of slender but not woody stems, and death of older leaves. Magnesium is absorbed by the plant in the form of magnesium ion. Magnesium is part of the structure of chlorophyll. It actually occupies the center of the porphyrin ring. It also has a role in the activation of enzymes involved in respiration, photosynthesis, and the synthesis of DNA and RNA. Magnesium is an activator of ribulose biphosphate carboxylase and also phosphoenol pyruvate carboxylase, two critical enzymes involved in carbon fixation. Symptoms of magnesium deficiency include chlorosis between veins and premature leaf abscission. Potassium is absorbed by the plant in the form of potassium cations. Potassium contributes to solute concentration of cells like stomata, resulting in its opening when potassium accumulates in the guard cells and its closure when potassium leaves the guard cells. Unlike other macronutrients, potassium is not structurally bound in the plant, but like nitrogen and phosphorus, it is highly mobile. Potassium also activates many enzymes involved in respiration and photosynthesis. In potassium, the deficiency symptoms first appear in older leaves, which characteristically develop mottling or chlorosis, followed by necrotic lesions, that is, spots of dead tissue at the leaf margins. Stems are shortened and weakened, and susceptibility to root rotting fungi is increased. The result is that potassium deficient plants are easily lodged or easily bent to the ground. Calcium is available to plants in the form of a divalent cation. It plays a role in the synthesis of new middle lamella. If you recall, the middle lamella is composed of calcium pectate. It also plays a role in the mitotic spindle during cell division. It is needed also for normal functioning of the plasma membrane. The plasma membrane becomes leaky, or the physical integrity of the membrane is compromised when there is insufficient calcium. Calcium acts as a second messenger to responses to environmental and hormonal signals. Calcium may actually bind to calmodulin, a protein found in the cytosol of a plant cell, and the calmodulin-calcium complex regulates many metabolic processes, such as the activities of a number of enzymes. Characteristic symptoms of calcium deficiency include general chlorosis, necrosis of young meristematic regions, such as tips of roots or young leaves, where cell division and wall formation are most rapid and, as mentioned, requires calcium. Severe stunting of the plant results if tips die prematurely. Also, roots become slippery to touch because of the deterioration of the middle lamella.